Assalamualaikum and good day. I bid to Dr. Yusuf and my name is Tasnim. Today I would like to present for the video presentation assignment. The topic is plant modification through genome editing technology. Firstly, we go to the introduction, precursor to genome editing. Decades ago, the hunger and malnourishment problem has become a global problem. By 2017, 821 million people classified as chronic malnourishment, with 150 million children have stunted growth. To strive for Sustainable Development Goal number 2, which is zero hunger, agricultural system needs to improve time by time to reduce hunger index and to meet food demand with increasing global population, which estimated to be 9 billion people by 2050. One of the advancements in agriculture technology is genome editing tool. According to Zhang et al. 2018, genome editing tool is a technique to modify organisms' genome precisely, modification at locus. And next one is the genome editing able to characterize gene in terms of function and improve the agricultural traits by adaptation to extreme weather and essential nutrient for example like golden rice, and any other commercialization traits like bigger size and sweeter taste. Several genome editing tools are zinc finger nuclease, transcription activator like effector nuclease, cluster regularly interspaced short palindromic repeats, which known as CRISPR-Cas9. Next slide is general mechanism of genome editing. Genome editing tool make use of sequence-specific nuclease (SSN), which recognize specific DNA sequences, subsequently produce double-stranded breaks (DSB). And then it will stimulate repair system in plant to fix the DSB. There are two pathways of genome editing, which is by non-homologous end joining and NHEJ, such as insertion and deletion of nucleotide, and by homologous recombination (HR), for example gene replacement or mutants insertion. See at the right picture. Firstly, we observe the target sequence, and after that, the guide RNA will bind to the target sequence, and then the Cas9 enzyme will bind to the guide RNA, and lastly, we'll introduce the mutation to the genome. Next slide is successful crop with genome editing tools. These are the crop species with the aims to improve its traits by using the genome editing tools. For example, maize, the gene editor is ZFN, and it aims to produce herbicide-tolerant and phytate-reduced maize. And these are the other examples that can be seen in this slide. Next slide is genome editing tools and DNA repair system. There are three types of the genome editing tools. Firstly is zinc finger nuclease, transcription activator like effector nuclease, TALENs, and lastly clustered regularly interspaced short palindromic repeats, CRISPR-Cas9. We move to the next slide. In this slide, I will briefly explain about the genome editing tools and the DNA repair system. Firstly, we go to the zinc finger nuclease. ZFN is a combination of zinc finger-based DNA recognition modules and the DNA cleavage domain of the FOK1 restriction enzyme. Individual zinc fingers will bind to nucleotide triplets, and the fingers will accumulate and produce chimeric nuclease, which then bind to the specific DNA sequence to produce double-strand breaks. Example of ZFN is PAT or PAT gene cassette was inserted to maize gene to produce the herbicide tolerance and altered inositol phosphate. Second genome editing tool is transcription activator-like effector nuclease TALENs. It is a combination of transcriptional activator-like effector repeats, TAIL, and the FOK1 restriction enzyme. TAIL repeats target single nucleotide and produce TALEN chimeric protein. TALEN chimeric protein will bind to the target sequence and produce indels mutation to the sequence. TALEN has large potential in target design and target site compared to ZFN because it targets single nucleotide and it was proven to give changes to phenotype. For example, TALEN has succeeded in destruction of bacterial blight susceptibility gene and produced rice with resistance to bacterial blight disease. The third genome editing tool is clustered regularly interspaced short palindromic repeats CRISPR-Cas9. It is the pathway of CRISPR-Cas9 is homologous recombination, and this system uses single guide RNA (sgDNA) to guide Cas endonuclease to the target DNA. And Cas9 nuclease needs protospacer associated motif (PAM) at the NGG downstream of the target site to work properly. Type 2 CRISPR SP-Cas9 system is taken from Streptococcus pyogenes, and it is much easy to use as it is more simple, low cost, and more efficient compared to ZFN and TALEN. CRISPR-Cas9 able to target multiple genes editing. Example of CRISPR-Cas9 is it able to modify 3 homologs of EDR1 to produce powdery mildew-resistant plant. In conclusion, to reach the goals in SDG, especially Zero Hunger Goal, scientists always work to produce more efficient gene editing tools to ease the process in producing desired characteristic crop. ZFN and TALEN are effective tools for genome editing, but these systems take more time and laborious method to produce desired results in modification. Therefore, CRISPR is introduced due to its efficiency, precise editing, and simple method to perform. Lastly, gene editing technology brings lots of benefits to humans as it enhances the crop nutrients, tolerant plant to pest or extreme weather, and produce higher yield production with good traits for the crops. Thank you for watching.